Hello, I Vani Singh. Today we will study about the reproduction in organisms, class 12 topic, chapter 1. What is reproduction? Reproduction is the ability of an organism to produce the young ones of its own kind. There is a period between the birth to the death. It is known as the lifespan. Birth and the death. Between them, it is known as lifespan. The lifespan can be studied in the various headings: birth, jubilant phase, reproductive phase, senescence phase. Senescence phase is also known as the aging phase, and the death. Different organisms have the different lifespan, maybe years, weeks, hours, or the many types. First, the shortest lifespan is of mayfly, having the lifespan of 24 hours. Tortoise, having the lifespan of 300 to 400 years. Bowhead whale, having the lifespan of 150 to 200 years. Parrots, having the lifespan of 140 years. Now we will study about the types of reproduction. Firstly, asexual reproduction and the sexual reproduction. Firstly, we will study about the asexual reproduction. In asexual reproduction, in this only one parent is involved. If the male is involved, then female will not be involved. If female is involved, then no male will be involved. In this, there is no fusion of gamete. And second, there is a mitotic division. In sexual reproduction, there is the formation and the fusion of gametes. Means in this, both male and female will be involved. In asexual reproduction, most of the times we use the term clone. What is clone now? The clone is when the children is means when the offspring is genetically similar to the parent. There is no differentiation. We can't distinguish that it is a child or the parent. Means they are looking both similar. In sexual reproduction is a variation and the recombination. In sexual reproduction, the offspring is genetically different from the parent. Now we will study about the asexual reproduction in plants. जो asexual reproduction in plants है, it occurs by the two methods, apomixis and the vegetative propagation. In apomixis, it occurs by the agamospermy. Now, what is agamospermy? Formation of an embryo from an unfertilized ovule. Means there is a formation on embryo which is an unfertilized. Means through the asexual reproduction. Apomixis can be further divided into three types: adentive polyembryo, recurrent agamospermy, and the non-recurrent. Now first we will study about the adentive polyembryo, in which embryo develop from the diploid cells of the nucellus. The nucellus is in mass and embryo is in diploid. Example, citrus plant. Next, recurrent agamospermy, in which embryo is in diploid, means 2n, which is developing from the megaspore mother cell. In this ovule, for example, there is an ovule and each cell of an ovule is developing megaspore mother cell. Next, we will study about the nucleant. In this, embryo is an haploid, which is an unfertilized egg. It is known as parthenogenesis. Next, we will study about the vegetative propagation. Vegetative propagation occurs by the various methods: stem, tubers, and rhizome. Now, firstly, we will study about the stem. In stem, the first example comes runner. If we will see the example of this is an example of a runner. There is an horizontal branch in which nodes and internodes are coming. These are the nodes and between this these are the internodes. This one is the root and upper one are the leaves. when these leaves are coming in direct contact with the soil, from that runner will develop. Next, offset. Example means they are the type of aquatic. Example, terror of Bengal. Example, Econia. See this one diagram. This is a horizontal branch. Nodes and internodes are there. Swollen part is there. When this swollen part come in contact with the soil, the new branch will develop. This is an example of an offset. Next, in tubers. Tubers in example is this potato. In potato we see the scars, and scars indicate there is an eye. In some tubers means potato, there are yellow growing plants which indicate that there is a origin of a new plant. Next, rhizome. In rhizome there is an example of ginger. This is an ginger. There are some lines. This one part is a node and this one part is an internode, from where new plants has been developed. The plants has been developed from the node portion, not from the internode. You can see this carefully. Next, root. Example, sesame and sweet potato. In root and from roots, the roots has been developed from the bud portion. Next, leaf. The leaf has also been developed from the buds portion. Example, bryophyllum and the bignonia. Mention one thing, the bignonia, bryophyllum, these all are botanical names. This should be underlined. Next, artificial propagation. Artificial propagation can be done by the various methods: cutting, grafting and budding. In budding, there is example of an amoeba, which is an immortal, which never dies. Next, grafting. In grafting, we take the two portions, means we take the two stems which we cut by this slant way, in which the characteristics of both the plants come. Cutting. In this stem is cut in slant way, so that which is cut below the nodes, so that the tissue cannot be damaged and it is digged in oxygen. With this end, we do the end of our topic, and I hope your all doubts have been cleared. Thank you.